Okay, this is our first test run in me recording my lectures and giving them to you online, so hopefully this works well. Today we need to talk about mutations and how they relate to protein synthesis. And what we need to start talking about first is, what do you think about when you hear the word mutation? A lot of you are probably familiar with comic books, and so you think of the X-Men, and you think of a mutation as being something really, really severe, or you might think of the Ninja Turtles as them being mutated and it being something extremely severe. So you probably have some kind of a either advantageous view of mutations, or you have some kind of a negative viewpoint of mutations. And so when you hear that, you think that it is something that is bad, or you think that it is something that is very, very severe. And what we'll talk about today is that is not necessarily the case. A mutation is a change in an organism's genetic information. And so it is some kind of a change in the DNA. And so we're taking something inside of this DNA sequence here and we're changing it. And what we've been studying is this idea of protein synthesis and how the DNA turns into messenger RNA, which will then come down here and turn into a protein for us. And so what we'll find out is that this mutation, or this change in the DNA, will then affect our protein sequence, or our end result that we're desiring. Okay, so mutations in protein synthesis. Here we see a normal example of protein synthesis. We see our DNA strand, we see a messenger RNA strand, and then the amino acid chain that comes from that. So note once again that the DNA is transcribed into messenger RNA, which is then translated into our amino acid chain. So if we're trying to review here, we would call this process transcription. And then this process of going from messenger RNA to amino acids is then called translation. Okay. And so if there's any change whatsoever in our DNA, then that is going to affect our messenger RNA, which will then affect our code for our amino acids. And so a change in the amino acid sequence could be a big deal, or it could be a small thing. We'll figure those things out as we go. All right, so let's take a look at an actual mutation. Okay, so at the top here, what I've done is I've used this exact same example from the previous slide. Okay, and then now down here I've shown you an example of a mutation in which we've changed just one nucleotide. Okay, so you'll notice that up here our sequence is TACGCA, but down here we've changed that C to a T. And so we've mutated this DNA. And so what that does then is that changes our messenger RNA, which in turn changes one amino acid. And this is one of our examples of a mutation, and we'll talk about what kind of a mutation this is in just a moment. So there's two main types of mutations. The first type is called a point mutation. Okay, point mutations. A point mutation is a mutation that occurs at just one point on the DNA, and so this is going to produce a change in a single gene or a single trait. The remainder of this lesson will cover point mutations because in Unit 7 that's really all that we're concerned with is point mutations. A second type of mutation is called a chromosomal mutation. This is a mutation at the chromosomal level, and so remember we talked about that, how DNA might look like this. But then we talked about how it winds itself up really tightly into this little shape that we called a chromosome. And so when the whole change, or sorry, when the mutation is on the chromosome, then that's what we call a chromosomal mutation. We'll talk about that whenever we get into genetics in Unit 8. All right. So the first type of mutation that we'll talk about, it's still a point mutation because it's just happening at one point, but it is called a substitution mutation. Okay. Now, when you hear that word substitution, I want you to think of a substitute teacher. There is still a teacher in the classroom, it is just a different teacher. So you didn't add a person, you didn't lose a person, you substituted a person. All right. And so if we take a look Substitutions only affect one nucleotide. The original nucleotide is substituted for a different nucleotide. All right. So here we can see our original DNA sequence is TACGCA TGGAAT, and that codes for these amino acids, or sorry, this mRNA, and then that codes for these amino acids. Here, what we're going to do is we're going to substitute one letter. The original DNA said GCA. Here, we're turning that into GTA. Okay. Now, what that's going to do for us is that changes one of the codons on the mRNA, which will then in turn change just one amino acid. Okay, kind of the easy way to remember a substitution, and one of the easy ways that we will refer to mutations is you usually use this sentence, the fat cat ate the rat. And so here we have little three-letter words that can represent our codons, and we can see the effect of if we just change one letter. And so here in our example, the original sentence we have, THE for the, but then if I change that one letter from an H to an E, I changed the sentence, but I didn't really make that big of a difference. Okay, so that's our substitution. The next type of point mutation is called an insertion. Okay, so just like it sounds, we are now inserting a new nucleotide. All right, so in an insertion, we have one additional nucleotide that is added to the original DNA. Now, what I want you to think about is I want you to imagine that someone is cutting in front of you in the lunch line. Okay, so let's go back to our sentence of the fat cat ate the rat. Here, we're not changing one of these letters. Instead, we're adding a whole new letter to that sequence. Okay, and what that does is it shoves every letter behind this H back one, just like that person that cut in front of you in the line pushed you back one further spot. Okay, now let's think about that in context of our DNA and our mRNA and then amino acids. Okay, so if this is our original strand up here, and down here we can see what would happen if an insertion took place. So here we inserted this T, and what that does is it pushed every letter behind it back one, and so that changes all of our codons. So you can see up here at the in our original, we started with the start codon AUG. Well, now that has been changed to AUA, which changed our amino acid. Okay, then the next codon was changed, which means that the next amino acid is changed. This codon is now changed because everything has been pushed back. That changes this amino acid as well. Okay, so that turns into a big problem now that somebody has cut in line, because all of these DNA nucleotides and then all of these mRNA nucleotides have now been shifted one letter back, and that changes every single amino acid in our sequence. Very similar to that, another type of point mutation is called a deletion. So just like it sounds, now we've taken one nucleotide and we have deleted it from the original DNA. So once again, imagine yourself in the lunch line, and now somebody in front of you just hauls off and leaves. That's a good change for you, bad change for DNA. So if we look at our original sentence again, the fat cat ate the rat, here that would be kind of like we just deleted altogether this H, and then we had to squish all of these letters together. So you can see that illustrated a little bit better right here, over here on the right. So here's the original sentence, the fat cat ate the rat. If we delete that one letter, we still have to have three-letter codons to code for amino acids, and so that's why we put these three letters together here. All right, but you can see that that does cause a problem. Okay, both insertions and deletions fall into the category of frame shift mutations. Okay, and we call them a frame shift mutations because these are the types of mutations that change every single amino acid after the mutation has taken place. Okay, a substitution that we talked about earlier will only affect one amino acid because you're substituting one nucleotide for the next. Frameshift mutations cause more of a problem. Okay, so if we kind of look at our example here, this is a substitution where all we've done is changed just this one amino acid because we changed one nucleotide on the DNA. Whereas down here, this is an example of an insertion. Here we've inserted this one extra nucleotide, which will then change every single codon, which will then in turn change every single amino acid after it. So that is an example of a frameshift mutation, and that's kind of a big deal. Okay. Now the next question is, are mutations harmful, beneficial, or none of the above? And the answer is that a mutation can cause no change at all. A mutation could be harmful, or a mutation could be beneficial. It just all depends on what kind of a mutation we're talking about here. Okay, so an example of a mutation with no change would be our substitution mutation. Okay, so we can take this sequence here, and then let's say that we change the DNA to, let's call this DNA, and let's say we change that sequence to TAC and then GCG. I'm going to shorten it and only use about half. That'll change our mRNA to AUGCGC. Okay, so you can see that that's different here. All that I did was I changed that letter, which then in turn changed this letter. So my amino acids here are still methionine, and then if you look at your codon chart, CGC still codes for arginine. So I didn't change my amino acids at all, even though this is a mutated letter. Okay, so that didn't really cause me any change. Okay, some mutations can be harmful though. Some of the diseases that we'll talk about that are the result of mutations include sickle cell anemia, Huntington's disease, let me highlight these, cystic fibrosis, something called PKU, hemophilia. We'll talk about all of these in class, but these are definitely harmful mutations. Now, there can be some mutations that are beneficial. In humans, there are some humans that have been found to have a mutation that causes HIV resistance. That means that the HIV virus cannot infect them. Also, by the time we get to a later unit over genetics, we'll talk about something called natural selection, and we'll talk about how some organisms can have minor mutations that cause changes that allow those guys to adapt to their environment, and if they adapt to their environment a little bit better, that can increase that organism's chance for survival, and so we would consider those a favorable mutation. Alright, the last thing we're going to talk about real quick before we're done here is, when does a mutation happen? Okay, mutations occur during DNA replication. Alright, so while DNA is trying to make a copy of itself, one of these nucleotides either inserts itself, one of them doesn't get copied, or one of them just matches incorrectly, and that causes one small change in our DNA. Now, what I want you to recall from previous unit is that DNA replication happens during the S phase of the cell cycle. So if you'll remember, we said that G1 is the growth stage, S is the DNA synthesis stage. This is where replication happens, so all of this is happening here. G2, we're growing a little bit more, then in mitosis we're creating 2 new cells. Okay, so that's our lecture on mutations. I'll see you in class if you have any questions.